you once again to this session. So in the last class, we discussed about the phylogenetic analysis using an open access software, Mega 10 or Mega X. So there we used the protein sequences pertaining to, myoglobin, so that we can have an idea about the evolutionary origin of myoglobin gene in different species of mammals. We'll be again repeating the practical that was done in the last class, but of course today we shall be taking a different approach. It's an alternative way of doing phylogenetic analysis, and today we shall take DNA sequences instead of protein sequence for phylogenetic analysis. So I think my desktop is visible to you.
Yes,
So
sir.
let me let me open the file that I've shared with you. There is a file I have also kept in my desktop. This is a notepad file. The sequence may appear differently in your computer, no problem. Here we are having one particular sequence from Japanese encephalitis virus, an isolate that we isolated in our locality during one of the projects that we had in the department. And this was of an envelope protein gene of Japanese encephalitis virus. So this particular sequence was also submitted to NCBI database, and they have provided us the accession number. And this is still there in the NCBI database. There are a lot of sequences deposited to NCBI database from our department. This is one of such Japanese encephalitis virus isolate envelope protein gene sequence. So there are several similar genes from the same virus, but there may be some mutations here and there in different isolates. And this is one that we are referring to for today's practical. So we would like to see that whether the sequence pertaining to the envelope protein of Japanese encephalitis virus isolate, which we obtained in our locality, we isolated it from pig, whether this is having high level of similarity to other virus isolates of the same region, or whether they are also similar to the similar virus which are in circulation globally. So we do not know that particular, that means part is not known unless we go for a phylogenetic analysis. So this is one purpose for which in molecular epidemiology, the sequences of viral genes, or for that matter, the conserved sequences of any pathogen can be compared with other sequences which are already there in the databases, so that we can correlate the properties of the pathogen with the isolates which have been already reported from elsewhere. Okay, so that we can also trace the source of origin of the isolate, or maybe we can also go for tracking it. That is very important in molecular epidemiological study of diseases. And therefore, today we shall take this task of having a phylogenetic analysis of this particular sequence with other sequences of the same virus, similar sequences of the same virus present in the online databases. So how will we approach? So we'll do the phylogenetic analysis using the same software, Mega X, but today's approach will be a little different. We will directly download the sequences of envelope protein of Japanese encephalitis virus isolates obtained from different regions or countries, and then we will compare it with our isolate. Okay. First of all, let us go to our isolate and see where it is, what are the properties, and then let us take it to Mega X. So for that, either we can copy the entire sequence without the name of the sequence, or a shortcut method is to take only the accession number, because as I said, in GenBank database, these accession numbers are unique identities of the different sequences. So they are unique identification number, so they are never duplicated or there is no any redundancy. So we will just select the accession number, not everything, MK6823H7.1. So this is the accession number of the particular sequence that we will copy. Control C. Now we will go to NCBI. We will go to NCBI, search NCBI in a browser, then go to the NCBI homepage, and there we will go to nucleotide database. Because it is a nucleotide sequence, we will select nucleotide database, and then we will search for this particular accession number. Paste the accession number in here in the search window. So this has been pasted. Now we click search. That means we are searching this particular sequence. So now it has been shown that this sequence is available. So this is Japanese encephalitis virus isolate ABT-CVK VJE002. So here you see ABT, Animal Biotechnology, College of Veterinary Science, Khanapara. Then VJE002 is our identification number. This is a laboratory identification number. We have the record from where we have isolated it, and its epidemiological data is already in our records. So if you see the details of the particular sequence, you see this is given here, the features of the sequence. The general features are shown first. When you see that this is the envelope protein of Japanese encephalitis virus, and the authors are so many authors. We were working in this particular project in a group in collaboration with the Institute of Bioscience, IBSD, Imphal. I forgot the full Institute of Bioresources and Sustainable Development. Sorry, Institute of Bioresources and Sustainable Development, Imphal, and then the Institute of Life Science, Bhubaneswar. So a team of workers, we did all these works together. So it was also published in a journal called Infection, Genetics and Evolution in 2020. So at the time when we submitted this to NCBI, this particular paper was in press, means it was not published at that point of time, but it was later published. So this particular sequence was derived from the samples which were obtained from our laboratory. Although the paper was written by so many people from different institutes under a particular project, Molecular Epidemiology of Japanese encephalitis virus in pig population of Orissa, Assam and Manipur states of India. But this particular isolate was authored by, this particular sequence was authored by us. Here it is written, Bora P, Borkolita LM, Rajkhua U, Dr. R, and Buraguhai L. This is from our laboratory. Okay, so this is the sequence. Why I'm showing this? You see how the sequence has to be read, and the general features and the other technical features of the sequence can be read by reading the summary of the sequence information provided by the database. Now, what we will do with the help of the BLAST software embedded in the NCBI website, so we'll do a BLAST first. As I already demonstrated to you in the last class, a BLAST is a basic locales alignment search tool. So when we use the term BLAST, it is also used as a verb. When we are going to BLAST it, means we are going to compare it by doing sequence alignment with the sequences of the GenBank database. So for that, this option is directly here. We need not have to open BLAST separately. Simply you will click here, run blast. Okay. So now this is the blast page. Now we will click blast. We will not change any parameter. Let it remain as it is, default parameters. Simply we will click blast. So it will take 2-3 minutes for getting the result. In the meantime, let us do one thing. Again, go for searching the particular sequence as we did in NCBI. The same sequence. We will take it now to our software, that is Mega 10. So the same accession number I'm searching once again to show you how it can be directly shifted to the particular software. Okay, so for that, you have to open the Mega X. Okay. So let us open Mega X. This is the Mega X front page. Okay. So now here we will start doing the alignment, then phylogenetic analysis. So for that, as we did in the last class, first of all, we have to go to align. Then select edit or build alignment. Then select create a new alignment. Okay. Then click OK, because this is a new alignment. It is asking, are you building a DNA or protein sequence alignment? It is a DNA sequence alignment. So we will click on DNA. So this is the alignment window we obtain. Here we have to bring the sequence. So for that, what we can do, unlike what we did in the last class, where all the sequences to be compared were loaded into a single notepad file, but instead of that, we'll directly download sequences from the databases. So for that, within this window, there is one option called web. You see, the fifth option in the menu is web. Click on web, then click on query GenBank. Query GenBank. That means you just ask some information by giving some input into the GenBank. That means you give the keywords and find sequences in GenBank. So click query GenBank. It is loading. So this page has come. This is pertaining to the GenBank search. So this is space where we are to give our input. So same sequence information will give the accession number. Then we will search here. So when we search it in this particular interface, you see the same information has come as we found it previously. The same information has been downloaded, okay, pertaining to the same sequence, because we are searching in the same nucleotide database of GenBank. But here in this, there is one additional option available here in the particular interface. It was not there in the original search. Add to alignment. On the top, you see, after the menu bar, in the address bar itself, it is showing one additional option given here, add to an alignment. Now, if you click add to alignment, the FASTA sequence of this particular accession number or the sequence will be automatically transferred to Mega 10. Okay, just simply you click add to alignment. So now it is asking, what do you want to import? Whether you want to import the CDS only the coding sequence or import full sequence. We will click import full sequence. Entire sequence will be transferring to Mega X. Import full sequence. Then we say OK. Click OK. Now it is asking what are the input labels sequence name or the label. So full information is given here below, you see, but it is asking what is the label that you want to use in the phylogenetic analysis. So for that, we will click on first word. Then any one of these, they have given 4 options to you. Japanese encephalitis virus, the particular laboratory identification number of the sequence, and then the species, swine, and then the accession number. All these 4 options have been given. We will take the second option because this is giving us the particular identification number from which we can identify that this is nothing but a local sequence that we want to compare. Okay, this is our sequence. So we have selected the second option. Then click OK. Now you see this sequence has appeared in the alignment window of Mega X automatically. Automatically it has come here. So now let us go back to our browser to see if the blast has been completed. So we go back to the blast and we see that the blast has been completed. Okay, that means our sequence has been aligned with thousands and thousands of sequences present in the GenBank database, and those showing highest similarity to our sequence have been shown here. You see, there are many envelope protein of same Japanese encephalitis virus, with which it has shown up to 100% similarity, 100% identity. So identity percentage is 100%. Then some are there which are having 99.74% identity. Some are there with 99.49% identity, like that. So gradually, if you go to bottom, you will find lesser and lesser identities. Say some are having 99.23, some are having 98.97, like that. So that means there are many similar sequences in the database which may be having similarity to the extent of 100% to 99% variation. Some mutations have been already there in some of these strains. So for doing this phylogenetic analysis, we will have to now download other sequences which are having similarity or dissimilarity with our sequence, so that we can finally draw a conclusion regarding the phylogenetic relationship among the isolates or the sequences based on their sequence similarity percentage. So for that, we will not download many sequences today. You can download as many sequences as you like, but today, so that we can complete the practical within short time, we shall only download 5 or 6 sequences to have a comparison. Okay, but again, if we take the envelope protein gene sequence, either partial coding sequence or the whole gene sequence, that will be better for us today, because alongside, if we also download some complete genome sequences which contains not only this gene but also sequences of various other genes of the virus, then it may take a lot of time to do the phylogenetic analysis, because the sequence length will be variable. Some sequences will be shorter, some sequences will be longer, because the genomic sequences will be very long. But we need only sequences of this particular envelope protein only, so that it is comparable. So our sequence, we know that it belongs to the envelope protein of the virus. So we need similar sequences, not the whole genome sequences. So we'll only download those sequences which are envelope protein gene sequence. Okay. Suppose we will download the number 1. Let us see number 1. First one is this one. So this is the accession number. You can directly copy this accession number or note it down from here. Okay, you can directly copy it like that. So just copy the accession number, nothing more. So just highlight it, then Control C, copy. Or you can note it down in a paper, then so that you can search it in the GenBank. Okay. Now we go to the GenBank database. Again, already we have this page or the window that we have not closed, so that we can search more. Earlier we searched our sequence. Now we will search this particular accession number by the same process. Nucleotide, this accession number, then we search. So if we search in the GenBank directly, I'm showing it once again. You see, you get the detailed information about this particular sequence. But interestingly, you do not have that particular option. That is, what is available in the mega aid to alignment, that option is not present here. Okay. So directly by searching in GenBank, you cannot find that aid to alignment option. Okay. For that you have to go to mega 10, and again you have to search in the web by going to the mega 10. So I'm showing it again. I'm closing it. Of course, already this particular window is present in my computer, but I'll close it to show you once again. Go to web. Where is GenBank? Here you have to search. Okay. Not directly in NCBI. You have to search by going into web, then query sequence. So query GenBank. Then here you are to put the accession number. Sorry, I think there is some problem with the download. So we'll go it once again, no problem. We'll go to web, then query GenBank. Let us wait for some time so that it gets downloaded. Okay, it is downloaded now. Now we will paste our accession number here. Okay, then you can drag it to the middle, you can click search, or simply you can enter. Click enter. Okay, now this sequence has appeared here. At the same time, we have this option aid to alignment. After it is downloaded, just click aid to alignment. Again, it is asking which one you want. Import full sequences. Click OK. Then go to first word. Select the identification number of the sequence, JEB Imphol. You see, this was another sequence from our own project, but this was from Imphol. Click it here. OK. So this is the second sequence. It has already come in the alignment window. So similarly, we'll go back to again the browser to go to the last window. Okay. Then again we will select another sequence. Let us take now another sequence having 99.7% similarity, not 100%. So we will select the accession number, copy, then go to directly mega X, go to the web window, then search it here. Click it here, then search. It has come. So now you click on aid to alignment. It is taking some time. Okay, it has not been completed. So we'll refresh this page. Let us see. So there is some problem. We'll close it, then come from web once again. Go to web, then query GenBank. Here we will search once again. Okay, now it has been downloaded. So now we'll click aid to alignment. Again, import full sequence. Click OK. Go to first word. Then click ABT CVK. So this is again another sequence deposited from our laboratory, but it is having some mutation. That means not 100% similar to the previous one. So click here, then OK. So the third sequence has been uploaded. Like that way, you go back to the browser, go to the last window, then select some other gene sequence, but it should not be a complete genome. It should be some gene sequence pertaining to the envelope protein. Let us take this one. This one is having also 99.49% similarity, 99.49%. So this you will select. Select the accession number, copy it, go back to MegaX, go to the search window, click it here. So there should not be any gap before the accession number. Then search. Okay. Then click Add to Alignment. Import full sequence. Click OK. Then go to first word. Select this particular number. Then OK. So the fourth sequence has come. Let us download another sequence, 5 sequence. Go back again to the browser. Go to the blast window. Then let us search for a sequence having less similarity. So next sequence, suppose we take this one, 98.97%. This is the envelope protein gene. So again, we will take this one. Control C, go back to Mega, go to the search window, click the accession number, paste it here, then search. Okay, it has been downloaded. Now click on Add to Alignment. Again, import full sequence. Okay. Go to first word. Then select some number. I think this is the identification number of the virus isolate. So click here. It may be some from some mosquito species. You see, some species of mosquito is given. Culex, Tritylor incus. So from this particular mosquito species, it was isolated. I think it is from some other country. We'll see where from this isolate was obtained by going into the sequence information in the database. So now we'll just, for doing the analysis rapidly, we'll only select the specific unique identification number of the sequence. Then click OK. So all the sequences have been uploaded. Okay. Some of the sequences are from our local laboratory. From our laboratory, 2 sequences, 1 sequence from Imphal, and 2 sequences we have downloaded from the GenBank, which we do not know where from they are. We may check it later on by going into the GenBank accession number and see the details there. So now we will first go for sequence alignment and then phylogenetic analysis. For that, this window is sufficient. So we'll close the search window. It is not essential now anymore. So now this window will be working. So let me maximize this window. So here 5 sequences are there, but there's listed 6. As I said previously, that although numbers are 6 here, but 5 are actually the sequences. The first one is blank. So that is not essential to be included in either the sequence alignment or during the phylogenetic analysis. So we will be selecting only the 2nd up to 6th. So first click on the 2nd sequence, then shift, then the last. This is one way. Or otherwise, what you can do, you can click anywhere in the sequence in one nucleotide, say the first nucleotide or the 2nd, then click Control A. So all will be selected. This is another way, but if you click by Control A, the sequence 1 is also selected. That is why either we have to delete the sequence 1 first, then select others, or otherwise better is to first select the 2nd one, which is a really sequence, then shift and click on the last. So all the 5 sequences are selected. Then we will do the phylogenetic analysis. First the sequence alignment. So after selection, we will go to alignment, then align by muscle. As we demonstrated in the last class, muscle is a robust software which can give you sequence alignment. So we will not change any parameter of sequence alignment, just click OK. So sequence alignment has been done. So now you see the last number 6 sequence is showing the nucleotide sequences at the beginning, or rather it is towards the end because the sequence is towards left. If you drag this particular bar, the bottom bar, then you can come to the first page. So you see, the last one is a longer sequence compared to the others. The last sequence, SCDJY01, this is longer than others. Others are smaller. And real alignment is within the sequences which are common to part of sequence which is common to all the sequences. That means the part of the segment of the sequence which is corresponding to the initial part of the number 6 sequence or the end part of the number 6 sequence is not available in other sequences. That is why there are all gaps. So this part is not common, only present in one sequence. So for doing the alignment, we should not use this part, this segment, because unnecessarily, wrongly, it will show lot of disparity or lot of variation with other sequences, because in other sequences there is creation of gap automatically. A gap means negative score in alignment. So therefore, it will be showing great dissimilarity with the number 6 sequence. So, but that is not true because the sequence segment is not available. That is why there is creation of gap. So what we will do, we will keep the common part and eliminate the non-common part, the segment which is present only in the 6th. So for that, what we will do, first we will come to the first in the beginning and then click on the first column in the sequence, just above the sequences. There is one written here, species oblique abbreviation. So there it is shown as steric marks. So in the first star, we'll click. So if you click on that, then the entire column is selected. Okay, that means the column containing the first nucleotides in each of the sequences is selected if we click on this star. Okay, then you are to drag this. By clicking, you are to drag it up to the column where there are gaps in some sequence. That means till we reach the part of the sequence which is common for all sequences. So this part can be deleted. So we'll simply click delete in our keyboard. So that part in the beginning has been deleted. Now we'll check similarly towards the end by dragging this bottom bar. So in the end also, a big part is missing in most of the sequences except the last one. So again, this part has to be deleted by the same way. So we'll click on the star above the uncommon part. So here, then from there we will drag it till end. But it may not be possible to drag it till end like this. So you can delete it part by part also, no problem, if it is too long. Or otherwise, you can do it like this, just drag it to the end of the sequence. Or one part you can delete, then again the next part, again the next part. That way also you can do, no problem. Let me show you. Suppose this part already selected, I will delete it. Then let us go to the end, see whether some more parts are there. Yes, more parts are there. So we have to select everything. So go back again. There's a big sequence in the last sequence comparison to the others. So we'll click again, and by the same way we'll drag it. I'll try to do it faster so that we can complete it within a short time. So let me delete it once again, then again come back to the sequence, then click here, then delete it. So because the last sequence is too long, that's why it is taking some more time. So entire part which is not common should be deleted. Okay, now it is deleted totally. So only we have kept that part which is common for all the sequences. But this has to be done only after doing the alignment. Before doing alignment, you should not do anything. After doing alignment, you should do it. Okay? Then, after this, we are to save this file by giving a name, save this session. So go to data, then click save session. It will automatically create a mass file, as I said in the previous class, .mas file. So let us keep it on desktop, then give a file name. Let us give it as a JEV underscore EP envelope 14, JEV envelope 14. So click save. So automatically it will be saved as a mass file on the desktop. Now this window is alignment window. This is no more to be used. So we will restore it to the previous size, or maybe you can minimize, no problem. Then we are going back to the original home page of MegaX. Now we will go for Phylogenetic. Click Phylogenetic. Before that, of course, we should have checked the sequence. So this was the file originally present. Let us check it. Because after sequence alignment, we have to see how much similarity exists across the different sequences, based on which we have to decide about the method of alignment. Now you see all these 5 sequences are showing high level of similarity, all true. Only 1 or 2 mutations are there here and there. Suppose here there is a mutation A to T. 4 out of 5 are having A in this position, 1 is having T. Okay, A to T mutation is there. Similarly, in some other places also, here see in the last there is G instead of A, all others are having A. So here and there 1 or 2 dissimilarity are there, but mostly they are identical. So we will go for the method that is called, what method will be following? The first one, we will take the maximum likelihood method. When there is a large scale similarity among the sequences, then we take maximum likelihood method. So click maximum likelihood. Then it is asking on which file you are to do the analysis. So automatically it is going to desktop, because my sequence is in desktop. But in desktop, JEV, EP, there are 2 files. One is the original notepad file. We should not select that, but we should select this one. This is the mass file. If you keep your cursor over it, it shows what type of file it is. Type mass file. You see, MAS. So double click on this. Now, is it asking, is it a protein coding nucleotide sequence? Yes, it is a nucleotide sequence. So we'll click yes. So it is automatically giving us the window showing the parameters of phylogenetic analysis. Here we shall have to check whether the required parameters are set for test of phylogeny. Test of phylogeny means validation by bootstrapping. So this is not shown, it is none. So we'll click here. Then after clicking, we will get the bootstrap method. We'll click bootstrap. Then by default, the bootstrap replication number is 500. You can increase this to 1000 if you need, but let us keep it at 5000 so that hurriedly we can do it. Then without changing any other parameter, we'll click OK. Okay. What happened now? The following taxa has no data available for the selected analysis. As I told, that sequence 1 is not having any nucleotide sequence, but this was by default present. Actually, out of 6, only 5 were the sequences that we know. Either we could have deleted it manually, or otherwise, at this point of time also, we should say it is asking, remove these data to allow the sequence to continue. You say yes. It is easier. Unless you delete it, this analysis will not be done. So it is giving a warning that do like to remove this taxa, which is not having any sequence. We will say yes. Or otherwise, in the data file itself, we could have deleted the sequence 1, which is not really the sequence, but it was by default present. So maximum likelihood method is being followed. It is one of the 4 different common phylogenetic analysis methods used when there is a large-scale similarity across sequences. So now it is doing the analysis randomly by picking up sets of sequence information. Up to 500 times it will do. It is showing below, you see, replicate number 463, 475. So it is increasing. Up to 500 times it will do. It has done. Then it has shown the phylogenetic tree. You see, this is the phylogenetic tree built on the data that we have given. So you can read it from here that out of the 5 nucleotide sequences, we saw that one of the sequences of our laboratory, ABT CBK VJE 002, is 100% similar. They are in the same line, same perpendicular line. No branching, same perpendicular line. So they are 100% similar to another sequence from IMPHOL. Okay. One more sequence which was uploaded from our laboratory is also having high level of similarity with the IMPHOL strain as well as the other strain of our laboratory, but there is little variation from others. So this length, this horizontal length of the branch indicates the variation. So some sequence variation is there. That is why it is showing a horizontal line of this length. Then this is also, these 3 sequences are also similar to another sequence that we downloaded from the database that was not from this region or from our laboratory. You can go back to the GenBank to find out where from this particular sequence was really deposited to the GenBank. Then all these 4 sequences are making one single clade. It is one single clade. But the other sequence, SCDJY01, is a sequence of the same gene, but which is showing high level of dissimilarity with the other 4 sequences. So this is making an almost outgroup, means this is not similar to a great extent to other 4 sequences. Other 4 are in the same, 4 are in the same clade. So by going back to the information in this GenBank pertaining to these particular isolates, we can say what were the locations from where all these sequences were reported and how they are correlated to each other. And that will be very useful for studying the molecular epidemiology and transmission pattern of the particular virus in different populations. How the virus has been transmitted from one place to the other or one population to the other. So only for rapidly doing it, we have selected merely 5 sequences, and that is why a final interpretation cannot be made on this basis. If you have downloaded large number of sequences, 25, 30 sequences from different localities having variability to different extent to the local isolate, then we could have finally interpreted about the molecular epidemiological pattern of the virus isolate that was obtained in our locality. So I think you could follow each and every step, and you can repeat it in your laptop now. So this is all about, I wanted to show you today the alternative approach in doing phylogenetic analysis by directly downloading the sequence information from the GenBank and then directly uploading to the Mega 10 software. So this is a simpler method and faster method. And of course, here you must have to have very good internet connectivity. You must have to be online for doing in this way. Otherwise, if you download the information and have a local file already generated, you can do this phylogenetic analysis completely offline also. Thank you very much.